Welcome everyone. So we'll start with the next lecture topic, which is about using Markovian models to for modeling different biological sequences. So in this particular lecture topic, we'll start with the discussion on Markov chains, and in the subsequent lectures, we'll go on to talk about hidden Markov models and its different algorithmic aspects, and then applications for modeling different biological sequences. So far we have seen different algorithms for aligning and modeling genomes. So we have seen different sequence alignment algorithms. We have seen local and global alignment, where we can, using these alignment algorithms, we can infer nucleotide level evolutionary events, whether those are insertions, mutations, or deletions. And then we can also see that using a particular sequence, we can do a database search. So with a query sequence, we can scan for regions that may have some common ancestry. So we can look for more homologs of a certain sequence and then find a family of sequences by doing a database search. And we have seen different algorithms for doing that. So we have seen BLAST and different variants of BLASTs. And then we have seen different algorithms for multiple sequence alignment. So one of the technique that we have seen is detecting different motifs. So these motif detection techniques, they actually try to find a recurring pattern that appears in multiple DNA sequences coming from different species. And then we have seen also multiple sequence alignment, a longer version of that multiple sequence alignment in terms of looking at a profile that defines a family of sequences. So we have recently looked at position-specific spore matrix and correspondingly the profile, which can actually define a family of sequences. So it can store position-specific scores for a family of DNA sequences. So the question we are going to ask now is that, can we apply these ideas to model DNA sequences? So if I have a particular DNA sequence and we don't know anything about it, can we actually come up with certain aspects and certain models that can tell us something meaningful about the DNA sequence? So let's say we have a new sequence of DNA. What can we do with this new piece of DNA? Of course, one option is to align it. We can align it with things we know about, so we can do a database search. So we have seen already existing examples of searching through the database, and this will give us related hits, lots of hits of related sequences. Now we can also align it with unknown things. So this part have not been covered in the class and will not be also covered in this particular course. So there are assembly algorithms or clustering algorithms where we can actually align a particular sequence with unknown sequences as well. Next option is to visualize it. So we can actually visually inspect the content of the DNA. Does it have a nucleotide composition which is very non-standard? So basically the frequencies of the bases are different from what we are supposed to see. Does it have certain k-mers which are existing in higher frequencies than the others? Is there any recurrent pattern that we observe in the DNA? And so on. And the third option is to model the particular sequence. So by modeling, we refer to the situation where we can make some hypothesis about the sequence, where how the sequence was generated, are there any common ancestor for the sequence, are there any particular functionalities associated with certain regions of the sequence, and so on. And essentially, by modeling it, we are trying to build a generative model. Generative model is basically a mathematical model or a term used to describe the models which can actually generate some data. So we can think of the sequence, DNA sequence, or a protein sequence as data that is being generated by a particular mathematical model. By doing this sort of generative modeling, or coming up with the hypothesis that a certain model actually generated the sequence, we can describe different functionalities of this DNA, of this new DNA sequence. And this will also help us to find sequences of similar type. Now, by type, here I refer to different properties of the DNA that can be inferred from this model. So we have different chunks of DNA which are very long, unlabeled. The question is, how can we label the different chunks of DNA, these unlabeled chunks? So given a DNA sequence, can we label different regions? And basically, we want to build this sort of models so that we can actually identify different regions of the DNA. And we can associate or basically annotate these regions. We can try to understand which of the region is exon, which is intron, which is maybe a promoter region, maybe intergenic region, or some other region which is classifiable using some sort of function. If we want to build this framework, then this sort of framework will give us a lot of ability regarding the DNA sequence. So the first will be able to emit DNA sequences of certain type. So what do I mean by that? So this is basically saying that if we have a model for annotating DNA regions, then we can try to understand or try to identify sequences of similar type. And this similar type of sequences can be actually generated from the model itself. So we have a model of the DNA, and using this model, we can generate more sequences, more DNA sequences of similar type. And of course, here we are not talking about identical sequence. We are essentially talking about preserving some properties for the type of the DNA that we are modeling over here. Also, this sort of framework gives us the ability to recognize DNA sequences of a certain type. So this certain type of DNA, we can call this as a state. So if our model has certain specific aspects that are actually generating certain DNA sequences, then we can assign that particular aspect as state. We can call that a hidden state of the model. And we can try to identify what hidden state is most likely to have generated the observations that we are seeing in the DNA. And at the same time, we can also look for the interplay between these hidden states of the model that actually generated the very long DNA sequence. And finally, this framework will also enable us to learn different characteristics of each of these hidden states. So this will be very useful in learning the generative models on very large data sets. And once we learn these generative models, we'll be able to classify unlabeled data. So from the given DNA sequence for which we already have some annotations available, so let's say for which introns and exons are already annotated, then we can use such sort of labeled data to train our generative models. And once we learn that model, then we can actually use that model to classify unlabeled data. We can try to annotate regions in sequences which are not annotated at all. So in other words, whenever we encounter a new DNA sequence, we can use existing models to learn or annotate regions and learn about the functionalities of the regions in that DNA sequence. Now, oftentimes when we are talking about these generative models, we are talking about probabilistic models. Now, why is it useful to have a probabilistic model for the biological sequences? So, of course, there are many reasons for it. First of all, biological data is oftentimes noisy. So it is not very deterministic. If you collect data from one source and then you collect data from different sources, there would be inherent noise that you will be seeing in the data. So this data will not be only technical noise, but there could be also some sort of biological variability in the data. At the same time, this probabilistic modeling techniques can be very useful in updating our previous knowledge that we already have about the biological sequences. So in the probability literature, this is called about learning from the prior. So let's say we already know, we have some prior knowledge about the biological sequence. We want to incorporate that or describe that prior knowledge in terms of some mathematical equations, and then that prior knowledge can be useful in can be really updated when we obtain more sequences of the similar kind. And when we are using probabilistic sequence modeling, then we are not limited to yes/no kind of answers. So oftentimes, when we are talking about certain questions regarding a biological sequence or a biological system, yes/no answers may not be all the time very useful. We also need to have certain degrees of belief or degrees of confidence. So these probabilistic models, they already come with associated with some sort of degrees of belief or confidence. So in that way, it is very helpful. So the tools that we are going to use for probabilistic sequence modeling, these are known as Markov models. So there are classic Markov chains, which I will introduce later. And then there is also extended models for Markov chains, which are called hidden Markov models. So we are going to look into these mathematical models and see how they can be used for modeling biological sequences. And this is a very classic example of a hidden Markov model. So you can see that this is a graph where we have three different nodes. So these circles over here are nodes, and these arrow marks from one circle to another, one node to another, these are called transitions or edges. So this is basically a graph structure. So graph consists of nodes and also edges or transitions between these nodes. So this is a particular hidden Markov model to describe the weather of a particular day. And you can see, so there are three basic nodes. This one is actually encoding the sunny weather. C is cloudy and R is rainy. And we also see different transitions. So if a particular day is sunny, we see a transition from sunny to being cloudy, and there is a number associated with this particular edge, which is 0.3. So this says that if a particular day is sunny, the probability that the next day will be cloudy is 0.3, and so on. So the basic understanding that you can get, or basic idea that you can get from this particular example over here, is that we are going to use these graph structured models. And basically this Markov chain and hidden Markov models are structured as graphs or represented as graphs. Excuse me. And these graphs will also have some probability distributions associated with this. So in other words, these are also known as probabilistic graphical models. Hidden Markov models are actually applied in different biological sequence analysis problems, and it has found very wide application in a range of problems. So for example, hidden Markov models have been used for predicting genes. So if we have a sequence, a new sequence, and we want to identify the genes inside that sequence, we can use hidden Markov models to annotate the regions first and then identify the location of a gene. Now we have seen different dynamic programming alignment algorithms for pairwise as well as multiple sequence alignment. Now, hidden Markov models can provide very good generative models for performing this sequence alignment. And this is an example of a hidden Markov model used for pairwise sequence alignment. We can also use hidden Markov models for building a profile for a sequence family. And we'll extensively study this part in another subsequent lecture. And this is an example of the type of hidden Markov model or the type of probabilistic graphical model we are going to use for building a profile for the sequence family. Base calling is an essential step when sequencing a piece of DNA, and when in case of base calling, hidden Markov models have been applied. And this is a particular model that was applied for base calling. Now the DNA sequencing errors, there are certain sequencing errors that can happen during DNA sequencing. These can also be modeled using hidden Markov models, and it has found many more applications, some of which includes the prediction of the secondary structure of protein or identifying copy number mutations and so on. So in other words, hidden Markov models are very important mathematical models that have been applied in various biological sequence analysis problems. So to start with, we are going to look at a very simple example, and we are going to use the Markov chains or Markov chain model to apply on this particular biological problem. We'll see how we can use Markov chain to model these problems. So the particular problem that we are going to look at is CpG islands. So in any genome, in eukaryote genomes, the frequency of this dinucleotide CG, it basically occurs in low frequency than it should be observed by chance. So for example, in case of human genome, the GC content is about 42%. So we should observe G and C with probability of 21% and frequency of 21% for each of them. So if we consider this as the expected frequency of the bases G and C, then the expected frequency that we should observe this dinucleotide CG is about 4.41%. But actually, we observe this with a frequency of only about 1%. So what is the reason that we see this type of lower frequency of this dinucleotide CG? So this is because naturally this nucleotide cytosine, this gets chemically modified by a method called methylation. So this cytosine will be methylated to methyl cytosine, and this methyl cytosine gets easily deaminated and gets converted into thymine. So this C to T conversion is very naturally occurring in the DNA. As a result, we are not going to observe CG at a frequency that we should observe or that is expected. So the observed frequency of CG is much lower than the expected frequency. And these methylated CpG residues are often associated with housekeeping genes in the promoter and exon regions. However, it is very interesting to note that there are some regions where this methylation process is suppressed. And this is mainly suppressed in specific short stretches of genomes. And this can occur around areas near the promoter or the start regions of many genes. And these regions where this methylation process is suppressed are also called CpG islands. So in these CpG islands, this dinucleotide CG appears relatively frequently compared to the overall genomes or the other regions of the genomes. So the question is, identifying the CpG island can be very useful in identifying different genes, right? So corresponding to this CpG islands, we are going to consider a particular problem, and we are trying to we are going to try to solve that using a Markov chain or Markov model. So here is the problem. Given a short stretch of genomic sequence, can we decide that this stretch of genomic sequence come from a CPG island or not? So this type of problem, solving this type of problem will be very useful in a particular situation where we are trying to distinguish between these two different regions, like CPG island and non-CPG island. So this is the problem that we are going to look at first. In order to solve this problem, we need a model that generates sequences in which the probability of a symbol depends on the previous symbol. So we have seen that CPG island occurs at specific stretches of the genome. So specifically, if a particular nucleotide is coming from a CPG island, it's possible that the symbol or the nucleotide before it also can come from the CPG island. And CPG island will end at a certain position, and after that the non-CPG island region will begin. So if we have a model that basically generates a symbol depending on the previous symbol, that will be very useful in modeling the CPG islands. Now, the classical Markov chain is a very good and appropriate model that actually can do this type of tasks. So how would we go about using this classical Markov chain? So we'll describe the Markov model or Markov chain model for the DNA. So this is a graph structured model, and this is the Markov chain model for the DNA. So what we have over here is 4 different nodes. These are also states, and each of these nodes or states correspond to each of the letters in the DNA alphabet. So we have A, we have C, G, and T. So these are basically saying that each of these states actually emit one of these letters. So this state A actually can generate letter A, T can generate letter T, and so on. And we can also see that within these nodes there are certain transitions which are denoted as these arrows. So this is basically saying that after G, a T can be generated, or after T, a G can be generated. So all these arrowheads are known as transitions. And with each of these arrow, there is a probability parameter associated, which denotes the probability of a particular base or nucleotide following another particular base. So these probability parameters are also known as transition probabilities. So again, just to simplify the concept, this is a Markov chain model for the DNA consisting of 4 different nodes. The nodes representing the states corresponding to the 4 letters A, T, G, and C, and the arrows are basically showing transitions which are associated with transition probabilities, denoting the probability of a certain base or residue following another residue. So let's see how we can use this particular Markov chain model to represent a particular DNA sequence. So let's say we have a sequence X, which is this sequence of length L is equal to 8. Now we already know that there are 4 different states and the transition probabilities are there. The question is, what do we mean by the transition probabilities? So this is how the transition probability is mathematically denoted. So it's denoted as this particular symbol over here. This subscript S and T is basically saying that this transition probability is associated with a particular position. So where we are observing at ith position of the sequence a symbol T, given that we know that already that the previous position was showing showing a symbol S. So here xi is basically denoting the ith symbol in the sequence. So for example, the last symbol is t. So the transition probability associated with the transition g to t will be denoted as a subscript g and t. So this is how the transition probabilities are defined. Now, as I said earlier, that this is a probabilistic model. So any sequence, any observed sequence that we say that have been generated from a Markov model will be also associated with a certain probability. And this is the equation that we are going to use for calculating the probability of a sequence. So the probability of a complete sequence, we can divide it into each individual positions. So this was a particular DNA sequence of length 8. So the probability will be shown as like this. So probability of xl comma xl minus 1 all the way up to x1, which is the first position. Now when we are writing a probability in this fashion, this is also called a joint probability. So the probability of observing S is the joint probability that we observe certain residues at certain positions. So for example, we observe A at position 1, another A at position 2, T at the position 8, and so on. So this gives us the joint probability. Now, in the probability theory, joint probability can be represented by a product of a conditional probability and a marginal probability. So this is the equation. So this is a joint probability of two events A and B, and this can be represented as a product. The first term is a conditional probability, where this basically says that this is the probability of observing A given that B is already observed or known, and it is multiplied by another probability, which is the marginal probability, which is the probability of the event B. Now if we actually apply this particular rule of probability, where joint probability is actually represented as a product of conditional and marginal, then we'll end up with this particular equation. So you can try to actually do it at home. You just have to apply this probability many times. So in the first step, let's say we are trying to say that the sequence or the residue that we observe at position L is dependent on all the other positions. So that will be denoted as this. So this is the conditional probability. So probability of xl given xl minus 1, xl minus 2 all the way up to x1. And that should be multiplied by probability of xl minus 1 comma xl minus 2 all the way up to x1. So that will be the marginal probability. And that marginal probability is again a joint probability. So we can subsequently apply the same rule again, and that will be further divided. So in the end, we'll end up with a product over multiple terms where we have lots of conditional probabilities multiplied by the marginal probability of observing the first symbol. So this is the probability of the sequence, whole sequence, that can be given from this particular model. Now, since we are using a Markov model which has a certain specific property, which is also known as Markov property. So this Markov property says that the probability of each symbol depends only on the previous symbol, but not on the entire previous sequence. So what is meant by that? So this is the mathematical notation for the corresponding property. So we have a probability, a conditional probability, where the ith symbol of the sequence, we are trying to model it with the symbols that we have observed before, that is from position 1 to all the position up to position I minus 1. Now Markov property basically says that xi only depends on xi minus 1. So if we already know xi minus 1, the previous knowledge is not at all useful. So we can say that this particular probability can be represented as probability of xi given xi minus 1. So the previous symbol is good enough to actually denote the current symbol or determine the current symbol with certain probability. Now, as you can see that this particular probability of observing xi given xi minus 1 is also the transition probability associated with the transition xi minus 1 going to xi. So connect this to this particular equation of transition probability. So here we see that probability of xi given xi minus 1 is the transition probability. So if we use this updated value in this particular equation where we are trying to find the probability of the sequence, then we end up with these values. So the first term actually becomes probability of xl given xl minus 1. The second term becomes probability of xl minus 1 given probability xl minus 2, and so on, all the way up to probability of x1. So then we have a pattern over here. So p probability of x1 other than the probability, this particular term, all these other terms denotes transition probability. So it becomes probability of x1, and then that is multiplied by a product over I is equal to 2 to L, and then we multiply the associated transition probabilities. So we can see that the probability of a sequence which is generated from a Markov model of DNA can be very easily calculated using the transition probabilities. In addition, we would also require the probability for the observing the first letter or first nucleotide. So we need something else apart from the transition probabilities. So to model the both ends of the sequence, we are going to associate two more states or add two more states in the Markov chain. So we already had the Markov chain for the DNA. Now we are going to add two more states, which are begin and end state, and these are basically dummy states. We can see that begin state can actually, after begin, so this begin state is actually modeling the start of the sequence, and it can transition to either A, C, or T, or G. Similarly, end state is actually modeling the end of the sequence. So any previous residue can end at this particular end state. So first we have this begin state, and this begin state will be useful for computing the probability of the first letter in the sequence. So we can say that the probability of observing a certain residue S or certain nucleotide S at the position 1 is given by a transition probability from the begin state to this particular state associated with the residue. So then we have ended up with another transition probability. So basically this makes the process very homogeneous. Similarly, we can also assign the probability of ending at a particular residue T by another transition probability, saying that there is a transition probability associated with ending in the end state from a certain previous state, which can be either of ATGC, and this is the transition probability associated with that. So now we have modeled all the necessary probabilities with the transition probabilities. It's important to note that oftentimes we do not model the end of the sequence in the Markov chains. We can say that, or we can basically assume that sequence can end anywhere. So the reason that we are actually adding this explicit end state is because we want to model a distribution of the length of the sequence. So if we get a new sequence, it can be of different length. It can be of length 100, 200, 1000, 100,000, or so on. So the distribution on this particular length can also be modeled using this end state. So essentially, we are saying that there is a probability distribution over all possible sequences of any length. And as we end the end state, this actually also helps us to get us the probability. And the distribution that it follows is basically decays exponentially over length. So if a sequence is much longer, then the probability associated with that will be also lower. So now let us go back to the problem that we started with. We started with the problem of looking at a particular sequence and identifying it, whether it came from CPG island or non-CPG island. So for that, we will need a training dataset. Now, what do I mean by a training dataset? So the training dataset refers to a set of DNA sequences for which we already know which are the CPG islands and which are not CPG islands. Such a training dataset will be used to derive two different Markov chain models. So we denote them as a plus model and a minus model. So the plus model will say that the sequence came from CPG islands, whereas the minus model is corresponding to the remainder of the sequence. So whenever a sequence is coming from any other region other than CPG islands, that will be modeled by this minus model. Now, of course, between these two different Markov chain models, what will differ is the transition probabilities associated with the transitions. Now we have to derive these transition probabilities for each of these models based on the training data set. Now the transition probabilities for each model is computed using this particular equation. So here I am showing you the transition probability calculation for the plus model. So this is the transition probability associated with the transition of S to T. So let's say if we want to compute the transition from C to G, so the probability associated with this particular edge, then we are first going to count the number of times that G is followed by C. And we are going to use only the sequences that came from CPG islands for counting this number. And in the denominator, we have all possible transitions. So whenever C is followed by any other residue, either G, T, or A, or C, we are going to count all those numbers and sum them up in the denominator. So this will give us the frequency or the probability that a particular symbol T is actually following S. So for example, G, the probability that G is following C will be given by the equation where we replace S by C and T by G. In the similar fashion, we can also obtain the transition probabilities for the minus model. Now here I am showing you two tables, and these two tables were calculated based on almost 60,000 nucleotides. Like it's actually calculated based on a number of sequences where there were 48 putative CPG islands, and based on that, these particular values were derived. So these are the probabilities, transition probabilities corresponding to the plus model, the CPG island model, and these are the ones for the minus model, the remainder of the sequence. Now here basically each row can be read as following. So here it says that these particular values are showing the probabilities with which A is followed by these four bases. So A followed by A probability is 1.18, A followed by C is 0.274, A followed by G is 0.426, and so on. Similarly, these are the probability values for the minus model. Now you can observe that the probability of G following C is smaller than probability of C following G. So the dinucleotide CG, it has lower probability as compared to the dinucleotide GC. And it is the same for plus as well as minus model. So in case of minus models also, the probability of G followed by C is lower than probability of C followed by G. There is another interesting observation that if you look at the probability for GC, G followed by C, that is the dinucleotide CG, we have for the plus model, the probability of observing CG is higher than in case of the minus model, which is expected because these sequences came from CpG island, so there is higher chance that we'll observe the dinucleotide CG. And this came from the remainder of the sequence where the frequency of observing the dinucleotide CG is much lower. So once we have obtained these models for discrimination, these can be used to compute the log-odds ratio. So you probably remember the log-odds ratio. So what we are going to compute is log of the ratio of the probabilities for the plus model and the minus model. And since we are computing these in log space, so it will eventually become a sum over all the positions from 1 to L, and the summation will be over the log of ratio of the transition probabilities in the plus model divided by the minus model. So this quantity over here is represented by this value beta of xi minus 1 to xi. And these are the log odds ratio associated with the transition xi minus 1 going to xi. Or these are also the log likelihood ratios. So here we are observing the table corresponding to the values beta, which are represented as bits. So this log likelihood ratios can be actually represented in terms of bits. That's what we are observing over here. Now this is an example of learning from this particular data set. So here on the x-axis we have bits, and we can see two different areas. So these particular sequences, the dark gray ones, are the CpG islands, whereas these ones over here, the lighter gray areas, correspond to the other regions. So thus we can use two different Markov chain models to actually identify whether a particular sequence came from CpG island or not. So this is where we are going to stop now. So we have seen a particular example where Markov chains can be very useful in modeling certain aspect of the sequence. Here, the property or the aspect being whether the sequence came from a CpG island or not. So in the next lecture, we'll continue with the extension of Markov models to actually look into hidden Markov models.